Hello everyone, myself Ishani Mandal from Microsoft Research Lab India. So today I will be going to present BERTChem-DDI improved drug-drug interaction prediction from text using chemical structure information. So this is the paper which has been accepted in the KNLP workshop AACL IGCNLP 2020. So today I will be going to present the paper. So this looks like an agenda. So first I'll discuss about what is meant by the drug-drug interaction. Next, I'll briefly explain the importances of drug-drug interaction prediction. I'll talk about the dataset that we have worked on, the methodology that we are proposing. Among the methodology, we have some classification, like we have a text-based classifier of the relation. We have chemical structure-based a pipeline. And finally, a joint approach. So this joint approach is our BERTChem-DDI, which we are proposing in the paper. I'll talk about the experiments conducted, finally some results and conclusion. So this is typically a brief picture of the drug-drug interaction. So we have two drugs which are administered to a patient or any person who is suffering from multiple ailments or multiple problems, and it's not clear whether their interaction of the drugs in their body will be beneficial or harmful, because whenever we go and get ourselves checked by a doctor, the doctor needs to know about the prescription. So why does he do so? Because he needs to know what are the drugs that we are currently taking in, and thereby he will propose another drug in order to understand the underlying mechanism of action of the two drugs or multiple drugs. So why it is important? First of all, we need to know the polymorphism effect. So what is this polymorphism effect? It's the concurrent administration of two or more drugs to an individual, which might lead to severe interactions. The interactions could even be some side effects, and there are some already existing statistics in which we have a detailed explanation of the side effects. And also there can be good reactions as well. So the drug-drug interaction, if categorized into, will get the side effects as well as the benefits. So what is the aim? We in this data-driven world, we are in urgent need of very good models which have a good predictive performance. So in this paper, we will talk about a method in which we'll have some instances, input instances, in which there will be two drug names tagged, and then we are trying to predict the drug, whether these two drugs interact with each other or not. For that purpose, we leverage an external information of the drug entities, which we are taking in the form of this miles architecture. Then after that, we'll show that this type of methods, which have already shown good performance in the existing literature, with using our method, it proposes a state-of-the-art performance on the dataset. So the dataset used is our DDI extraction 2013 shared task corpus. So it's a relation classification type of corpus in which we have 5 relation labels. One is the mechanism, the effect, advice, int means interaction, and other. So basically the task is a relation classification task, a softmax layer which will either predict one of these 5. In this statistics table, what we are trying to show is that we have a distinct separation of the training and the test sets, and we want to know how many drugs and the normalized drugs we can get. So this is the overall statistics that you can also refer to in the paper. Next, we'll talk about the architecture. So this architecture was basically an already existing architecture proposed by Wu and He in 2019, in which their model tries to predict the entity level and the sentence level information, and using that information they are trying to predict whether they are interactive or not. Like, even if they are interacting, what type of interaction it is. So basically we have this entity level information, and the entities are tagged with the entity markers. Like here you can see the dollar and also the hash. So we have two different entities marked within the sentence. Then using the CLS token, you were trying to get the BERT-based contextual embeddings. So we have the mention pool average embeddings of the two different entities, and also a CLS output. So the CLS output is the H0. This is the HI and the HA are the average contextual average embeddings of the entity 1, whereas HK and HM are the average entity representation of the entity 2. And after that we are trying to get the fully connected and an activation layer over it. So in the paper you will get the details of the different notations, and also after we get these three different information from the sentence and the entities, we are concatenating them in a single output. And after that we are trying to use a fully connected followed by a softmax layer, which is ultimately our prediction layer. So this is the prediction that we are trying to do. Next is the chemical structure of drugs. So as we were telling that in not only the entity level information, we are also trying to leverage the chemical structure information of the drugs. And this is the name of the drug followed by its drug bank ID, and this is the chemical structure representation of the drugs, which is encoded in this format. So this is the SMILES representation. What is this full form? It's like a simplified molecular input entry system. So this is the final canonical representation of this 3D structure. So all the drugs which are present in the drug bank having a drug bank ID has a chemical structure representation encoded in the SMILES. So we are using a chemical structure variational autoencoder, in which we are trying to capture the sentence low dimensional representation of this SMILES input. So the working principle is exactly the same as in the variational autoencoder. So we are trying to get this information out from the chemical structure. And after that we are proposing this model. So what does this model look like? So suppose this is our input sentence, in which glypafloxacin and theophylline are the two drugs. These are the two drug entities. So we are doing a normalization. So what is this normalization? Is an entity linking, in which we are trying to query this name in the drug bank database, which will give me a drug bank ID and also its SMILES representation. And the SMILES representation, as I was talking about, so it is a canonical representation of the glypafloxacin drug, and similarly for the theophylline. So after we get that, we try to obtain these two representations from the low dimensional representation of chemical VAE. And after getting these two representations, instead of only taking care of the sentence level and the input entity information, we are also augmenting this chemical structure representation during the training process. So in the final layer, instead of concatenating these three outputs, we are also concatenating the representation, and that is also followed by a fully connected and softmax layer. So this is the main difference that we are proposing in our paper. And during pre-processing, we'll do an entity linking to the drug bank, then we fetch the SMILES representations, and then the drugs which were not normalized, which could not be normalized to drug entities, we are initializing them with a domain specific what to wait. So for the details, please refer to the paper. And we also aim to use these four representations, contextual representations, from the sentence level. So during the results analysis, we are doing three kinds of ablation study. So one is the how much the different contextual embeddings have this effect from the textual corpus. Then we'll look into how much this chemical structure representation helps after augmenting in our proposed method. And also finally we'll compare with the existing baselines which are proposed in the literature. So first of all, we can see from this study, from this ablation analysis, from only the textual corpus, using only the textual information and the entity specific information, we are getting this is the test set macro F1 score. So the BioBert PubMed version is performing the best, and since we have seen that these BioBert representations are doing well, so after we augment the chemical structure representation, we are also analyzing how much the performance difference is. So we can see that the macro F1 steadily increases as we keep on appending the chemical structure representation. So this is a quite interesting thing which we have explored in the paper. And also finally we compare our method with the existing models starting from 2017 to 2018, then 2019, followed by the recent method. So we can see that our method is steadily performing excellently well on the mechanism type of interactions. And also finally, when we are comparing it with the total representation, we can see that our method performs really very well, and it is giving us a state of the art performance on this data set. So thank you.